350 people. That is roughly the number of people who fit into a small wedding hall, a single large lecture theater, or the morning rush on one city bus route. Now take that number and sit with what it means. Nearly every one of the 10 to 11 million Ashkenazi Jews alive on the planet today carries DNA that traces back to a founding population no larger than about 350 souls. They lived roughly 700 years ago somewhere in a tight-knit cluster of medieval Central European towns, and from that tiny group, an entire branch of world Jewry exploded outward into the modern era. The number is so staggeringly small relative to the population it generated that it sounds like a mistake. It is not a mistake. It is a measured, peer-reviewed, statistically modeled fact pulled from the genomes of living people, and it sits at the center of one of the most contested questions in all of human history. Are Ashkenazi Jews the biological descendants of the ancient Israelites who once walked the hills of Judea and Samaria, who built the first and second temples, and who were scattered to the winds by imperial Rome? Or, as a persistent alternative theory insisted for nearly 2 centuries, were they something else entirely? A population of Turkic converts from the medieval Khazar kingdom whose ancestors had never set foot in the ancient Near East. The question is not obscure academic trivia. It reaches into modern political claims about identity, belonging, and the deep history of the land between the Jordan River and the Mediterranean Sea. And ancient DNA has now answered it with a level of specificity that has shocked even the researchers who ran the numbers. The old story, the one you could still find in thick history books and encyclopedias printed well into the 20th century, went something like this. After the Roman legions crushed the Jewish revolt in 70 CE and flattened Jerusalem along with its second temple, the surviving Jewish population scattered. Some stayed in the land, but a great many moved outward, first into the Mediterranean basin, then deeper into Europe. By the early medieval period, a distinct Jewish cultural sphere had crystallized in the Rhineland, the region of Germanic-speaking Central Europe that produced the term Ashkenaz, a word drawn from medieval Hebrew geography that originally pointed to the lands around the Rhine. These Rhineland Jews developed their own customs, their own liturgical melodies, and eventually their own Germanic language, Yiddish. Over centuries, persecution and economic pressures pushed them eastward in waves, into Poland, Lithuania, Ukraine, and the Russian Empire. By the late 19th century, a vast and culturally prolific Ashkenazi civilization stretched from the Baltic to the Black Sea, and its children would soon scatter again, this time across the Atlantic to the Americas and outward to every continent. The story was coherent. It described a people who preserved their ancient religion and their ancient texts while moving through the geography of Europe. What it did not describe, with any precision, was their biological relationship to the people who had originally lit the menorah in the temple in Jerusalem. For a long time, that part was simply assumed. The assumption was that the chain was unbroken, that these Jews of Poland and Ukraine and Germany were the direct genetic descendants of the Judeans who had watched Roman siege engines roll through the Kidron Valley. That assumption was never tested at a molecular level until very recently. When it was finally tested, it did not just survive. It sharpened into something far stranger and more detailed than anyone expected. But before we reach that sharpening, we have to walk through the counter-theory that tried to slice the chain altogether, because the shadow of that theory is what made the genetic findings so explosive when they finally hit the journals. The Khazar hypothesis. You may have heard the name whispered in certain corners of the internet, or maybe you read Arthur Koestler's 1976 book, The 13th Tribe, which popularized the idea for a mass English-speaking audience. The claim, in its boldest form, was that the Ashkenazi Jewish population descended not from the ancient Israelites, but primarily from the Khazars, a confederation of Turkic-speaking tribes who built a powerful kingdom in the Caucasus and the Pontic steppe between roughly the 7th and 10th centuries CE. The twist that made the story magnetic was that the Khazar ruling elite, or at least a significant portion of the Khazar population, converted to Judaism in the 8th century. The conversion is a documented historical fact, attested to in medieval sources, including the correspondence of a Khazar king and a Jewish dignitary in Muslim Spain. The Khazar hypothesis then takes a speculative leap, arguing that these Khazar converts became the demographic seed of the entire Ashkenazi Jewish population. Under this theory, the Jews of the Rhineland were not the children of Israelite exiles who had wandered through Rome and Italy. They were the heirs of steppe horsemen and converted traders who had never seen the hills of the Levant. The political implications were immediate and have been wielded for decades to buttress arguments that modern Jews, and particularly Ashkenazi Jews who founded the State of Israel, have no ancestral connection to the land. The hypothesis sounded seductive. It had a coherent historical engine, a dramatic plot twist, and it unsettled a foundational narrative. But it had one enormous problem. It was, as the genetics would eventually reveal, not slightly incomplete. Structurally wrong. In 2010, a research team led by Behar published a landmark paper in the journal Nature, titled The Genome-Wide Structure of the Jewish People. The study was not a small pilot project. It analyzed the full genomes of 121 individuals drawn from 14 different Jewish communities scattered across the globe, from Ashkenazi populations in Europe and North America, to Sephardic communities in the Mediterranean basin, to Mizrahi communities who had lived in the Middle East and Central Asia for millennia. The data spat out a pattern so consistent that the researchers could see the deep structure of Jewish population history glowing in their scatterplots. Read that sentence again. All of the major Jewish diaspora groups, communities that had been separated from one another by geography for roughly 2,000 years, clustered together genetically. They shared a common biological origin, and that origin was not in the Caucasus Mountains or the Turkic steppe. It was in the Middle East. The Ashkenazi genomes did not drift toward Turkic populations or cluster with modern populations in the Khazar region. They pulled, like a compass needle, toward the Levant. Behar and his colleagues explicitly addressed the Khazar hypothesis in their analysis and found no significant Khazar genetic contribution to the Ashkenazi genome. 0. A later study led by the same group, published in 2013 in Human Biology under the title No Evidence from Genome-Wide Data of a Khazar Origin for the Ashkenazi Jews, drove the final nail into the hypothesis. A single renegade paper attempted to revive the Khazar claim in 2013, using cruder methods and selective data, but the broader community of population geneticists reviewed it and found its conclusions unsupported. The consensus today is as close to settled science as population genetics gets. The Khazar contribution to Ashkenazi genomes is negligible to non-existent. The Ashkenazi Jews are not a Turkic people who adopted Judaism. They are a Middle Eastern people who adopted Europe. But what does Middle Eastern mean at the level of individual base pairs of DNA? In 2014, a team at Columbia University, led by Shai Carmi, published a study in Nature Communications that zoomed the microscope in to a molecular resolution no one had previously achieved. And what they found inside the Ashkenazi genome read like an ancient demography textbook written in code. They sequenced the complete genomes of 128 Ashkenazi Jewish individuals and used sophisticated statistical models to reconstruct the demographic history of the population backward in time. The first number they pulled out made everyone stop. They estimated that the entire Ashkenazi Jewish population today descends from a founder bottleneck of roughly 250 to 420 individuals who lived between 25 and 32 generations ago, which places them squarely in the medieval period, roughly 600 to 800 years before the present. Sit with what that number means. At some point in the late medieval era, perhaps under the shadow of a specific wave of persecution, perhaps due to a combination of migration patterns and social isolation, the effective breeding population of what would become the world's largest Jewish diaspora was winnowed down to the size of a single village. Everyone alive today who identifies as Ashkenazi Jewish, all 10 to 11 million of them, from a cardiologist in Tel Aviv to a novelist in Brooklyn to a mathematician in Buenos Aires, carries the concentrated genetic legacy of that tiny founding group. That bottleneck left fingerprints. When a population passes through a genetic bottleneck, rare mutations that happen to be present in a small number of founders can become dramatically amplified in subsequent generations. The Ashkenazi genome carries a catalog of such amplified mutations, and they manifest as a set of devastating recessive genetic diseases that occur at dramatically elevated frequencies. Tay-Sachs disease, a fatal neurodegenerative disorder that strikes in early childhood, appears in roughly 1 in every 30 Ashkenazi carriers, compared to about 1 in 300 in the general population. Gaucher disease type 1 has a carrier frequency of about 1 in 15 among Ashkenazi Jews. Specific founder mutations in the BRCA1 and BRCA2 genes, mutations that dramatically elevate the risk of hereditary breast and ovarian cancer, run through the Ashkenazi population at rates that have made genetic screening a standard part of medical care in Jewish communities. Familial dysautonomia, a disorder of the autonomic nervous system that affects the ability to swallow, cry, and regulate body temperature, is almost exclusive to Ashkenazi Jews. Canavan disease, Bloom syndrome, Fanconi anemia. Each of these is a distinct molecular scar left by the bottleneck, a specific spelling error in the genetic code that happened to ride along in those 350 founding individuals and then rippled outward across 10 million descendants. If you carry one of these mutations, you are not just carrying a disease risk. You are carrying a biological timestamp that marks your ancestry back to a particular medieval village in the Rhineland. The diseases are not the whole story of the bottleneck, but they are the part that makes the history viscerally real. You can read about population bottlenecks in a textbook, or you can hold a newborn baby and understand, in a way that no data table can convey, that the same historical compression that gave rise to a vibrant global civilization also left this particular gene spelling error in your family line. But where did these 350 founders come from? That is the question that weaves directly into the ancient DNA revolution, and the answer is more layered than any simple phrase can capture. The Carmi study also estimated the ancestry composition of the Ashkenazi genome, and the numbers it returned form a kind of genetic double helix, two strands wound together inseparably. Roughly half of Ashkenazi ancestry traces to a Middle Eastern source population, specifically to the Levant, the Eastern Mediterranean coastal region that encompasses modern Israel, Palestine, Lebanon, and Western Syria. The other half traces to European populations, with a particularly strong signal pointing towards southern Europe, especially Italy. Let that sink in. The Ashkenazi genome is not a simple thing. It is a 50-50 fusion of two ancient worlds, a biological record of a migration that began in the Levant and then intertwined with local European populations over many centuries. This is not a metaphor. It is a measurable proportion of ancestry, visible in the patterns of single nucleotide polymorphisms scattered across the 23 human chromosome pairs. And if you zoom in closer, this 50-50 split begins to tell distinct stories depending on which line of inheritance you follow. A 2013 study led by Marta Costa in Nature Communications, titled A Substantial Prehistoric European Ancestry Amongst Ashkenazi Maternal Lineages, focused specifically on mitochondrial DNA, the small ring of genetic material that passes exclusively from mother to child. What Costa's team found was striking in its asymmetry. Roughly 80% of Ashkenazi mitochondrial DNA lineages trace back to European origins, specifically to women who lived in Roman-era Southern Europe. The implication, pieced together by historical geneticists, is that the founding population included Jewish men who migrated from the Levant during the Roman period and in subsequent centuries, and that these men often married local European women. The children of those marriages were raised within Jewish communities, and their descendants carried European mitochondrial DNA forward through the female line, generation after generation, until it became the dominant maternal signature of the entire diaspora. On the paternal side, the story inverts. The Y chromosome, passed exclusively from father to son, tells a very different tale. A large proportion of Ashkenazi male lineages trace back to the Levant, and specifically, in many cases, to a set of Y chromosome markers that have an almost literary resonance. In 1997, a research team led by Karl Skorecki published a study in the journal Nature that became instantly famous because its findings read like something out of the Book of Exodus. Skorecki, a nephrologist at the Technion in Haifa, had been sitting in a synagogue service when he noticed that a man called up for the priestly blessing, a man who self-identified as a Cohen, a descendant of the biblical priestly caste that traces its lineage to Aaron, the brother of Moses, looked physically quite different from Skorecki's own Sephardic family. Skorecki wondered, on a hunch rooted in genetics, whether men who claimed Cohen descent across divergent Jewish communities might share a common Y chromosome signature. His team collected DNA samples from Jewish men who identified as Kohanim, both Ashkenazi and Sephardic, and from Jewish men who did not identify as Kohanim. The results were unambiguous. A specific set of Y chromosome markers, which came to be known as the Cohen modal haplotype, appeared at elevated frequency in both Ashkenazi and Sephardic Kohanim, at a rate of roughly 50%. The haplotype was far rarer in non-Cohen Jews. This means that a significant fraction of men who carry the oral tradition of descent from the biblical priesthood share a common male ancestor who lived somewhere in the Levant thousands of years ago, long before the Roman destruction of the temple sent the Jewish population scattering across the globe. The Y chromosome signature of the ancient Israelite priestly caste is alive and well in the genomes of modern men who get called to the Torah in synagogues from New York to London to Jerusalem. And the story does not stop there. The same Cohen modal haplotype was later found in an unexpected place, thousands of miles from the Levant, deep in the interior of southern Africa, among the Buba clan of the Lemba people. The Lemba have an oral tradition that claims descent from ancient Jewish ancestors who traveled across Africa from the Middle East. The genetics confirm that oral tradition. The Y chromosomes of the Lemba priestly clan carry the same markers as the Cohen men of Jerusalem and the Rhineland. If the story stopped here, it would already be powerful enough on its own. But ancient DNA had one more secret waiting. To understand how the Ashkenazi genome connects directly to the world of the Hebrew Bible, we need to go back to the bones themselves. In 2020, a team led by Lily Agranat Tamir published a study in the journal Cell that made the earth of the Bronze Age Levant speak. The researchers extracted and sequenced DNA from the remains of 73 individuals who lived in the southern Levant between roughly 4,000 and 3,500 years ago, a period that spans the Middle and Late Bronze Age, the era during which, according to the archaeological and textual record, the Canaanite city-states flourished, and, eventually, the people who would be called Israel emerged from the broader Canaanite cultural matrix. These were not just any ancient bones. They were the remains of people who lived in the very land where the biblical patriarchs, judges, and kings would later walk. Individuals who lived in cities like Megiddo and Tel Hazor, places that still bear their ancient names. The genetic data from these Bronze Age individuals was then compared to the genomes of modern populations across the Levant and the Jewish diaspora. The result was a direct, measurable genetic continuity. Modern Jewish populations, including Ashkenazi Jews, share a substantial proportion of their ancestry with these Bronze Age Canaanites, on the order of 60 to 70%, depending on the specific modeling approach. Think about what that actually means. The DNA of people who lived in the land of Israel 3,500 years ago, during the period when the stories of the patriarchs were taking shape, is still detectable as a major ancestral component in the genomes of modern Ashkenazi Jews, living today in New York, London, and Tel Aviv. This is not a theological claim subject to faith or a literary claim subject to interpretation. It is a measured, quantified, statistically significant genetic relationship visible in the base pairs of the human genome. The books of Judges and Kings, the poetry of the Psalms, the laments of the prophets. These texts describe the lives and struggles of a people whose direct genetic descendants are alive right now, carrying that same biological inheritance into modern cities and modern lives. But the Bronze Age Canaanite ancestry is only one strand of the braid. The genetic story of Ashkenazi Jews is not a story of pure descent. It is a story of mixture, of movement, of human lives crossing boundaries that the textbooks sometimes imagine as hard walls. A 2017 study by James Shuey and colleagues published in the journal Genetics under the title The Time and Place of European Admixture in Ashkenazi Jewish History refined the timeline of how and when the European half of the Ashkenazi genome entered the picture. By analyzing the length of shared chromosomal segments between Ashkenazi Jews and European reference populations, the researchers were able to estimate not just what proportion of ancestry came from Europe, but when the admixture events occurred. Their models pointed to a window roughly 24 to 49 generations ago, which translates to approximately 600 to 1,300 years before the present. The geographic signature of the European admixture pointed strongly toward southern Europe, specifically Italy. This makes historical sense. The Jewish diaspora first took root in significant numbers in the Roman world, particularly in Italy and the broader Mediterranean basin, in the centuries surrounding the turn of the Common Era. Jewish communities are attested in Rome, in the port city of Ostia, and in cities across the Italian peninsula. It was here, in late antiquity and the early medieval period, that the genetic merger between Levantine Jewish men and local European women took place, producing the mixed ancestral population that, generations later, would move north across the Alps into the Rhineland and form the Ashkenazi core. Picture the human reality behind the statistics. Try to imagine a specific man, sometime in the 2nd or 3rd century CE, a Jewish merchant or a former soldier or a craftsman, whose father's family had lived for centuries in the hills of Judea. Perhaps his own father or grandfather had seen the temple in Jerusalem before it fell, or perhaps he carried only stories of it, told over oil lamps in Roman insulae. He settled in a port city on the Italian coast, where the Mediterranean slapped against stone quays, and ships arrived daily bearing olive oil, wine, and news from the eastern provinces. He met a woman there. She was Italian, a local, her family rooted in the peninsula for countless generations. Her mitochondrial lineage, one of the countless deep branches of the European maternal tree. They married. Their children grew up speaking both the languages of their parents and moving between the cultural worlds of Roman Italy and Jewish tradition. Multiply that single couple by hundreds, then thousands, across generations. Their descendants moved north, into the foothills of the Alps, then across the mountains into the green river valleys of the Rhineland. By the time the Ashkenazi bottleneck squeezed the population down to a few hundred founders in the medieval period, those founders already carried within their genomes the twinned ancestry of the Levant and Italy, the Temple Mount and the Roman Forum, the poetry of the prophets and the languages of Latin Europe. The genetic data tells you that this is not a romantic reconstruction. It is a description of molecular events written in the DNA of living people. If your family carries Ashkenazi roots, the specific mixture in your own DNA right now reflects this layered history at a level of detail that would have been unimaginable to your ancestors. In your autosomal chromosomes, the 22 pairs that recombine each generation, you carry segments inherited from Bronze Age Canaanite farmers who tilled the valleys of the southern Levant. You carry segments inherited from Italian women in the Roman world who contributed their mitochondrial DNA to the matrilineal line. You carry segments that survived the terrifying squeeze of the medieval bottleneck, passed through the hands of parents in the Rhineland who buried children lost to plague or persecution, and who kept going. You carry segments that traveled east in the Great Migration that filled the shtetls of Poland and Lithuania and Ukraine with Jewish life. And if you are a man who carries the Cohen modal haplotype on your Y chromosome, you carry a paternal lineage that has remained remarkably stable across 2,000 years of diaspora, a biological thread that connects you directly to a specific priestly caste that once served in the temple in Jerusalem. This is not a metaphor. This is not a poem. It is biology. Read through the lens of population genetics. Stop and think about what that actually meant for a moment. The same historical forces that scattered a people across continents and languages and centuries did not scatter them in the genome. The genome remembers. And here is where the story turns. Because the genetics does not only confirm connection, it also complicates it in ways that matter deeply and that require honesty. In 2016, a landmark study led by Iosif Lazaridis and published in Nature, titled Genomic Insights into the Origin of Farming in the Ancient Near East, sequenced DNA from Neolithic and Bronze Age individuals across the region and compared these ancient genomes to modern populations. The study confirmed that multiple waves of migration and mixture have shaped the modern Levantine gene pool, and that no modern population is a pure descendant of any single ancient group. The same is true for the Ashkenazi genome. The Bronze Age Canaanite ancestry is real and substantial, but it is not the totality. The European admixture is real and substantial, but it too is not the totality. The Ashkenazi genetic identity is a composite, a mosaic, the product of specific historical events that brought together populations who had previously been separated by geography and culture. This is not a weakness in the science. It is science being honest about what it can and cannot claim. The DNA evidence decisively refutes the Khazar hypothesis and confirms the Middle Eastern origins of the Ashkenazi population. That much is clear. But it also refutes any simplistic narrative of pure, unbroken, unadmixed lineage stretching back to biblical times. The Ashkenazi Jewish people are, in genetic terms, a Middle Eastern people who, over the course of their diaspora, incorporated substantial European ancestry. They are the children of both Jerusalem and Rome, both the Levantine hills and the Italian peninsula. The truth is richer and more human than either extreme of the political debate ever allowed. The political implications of this research hang heavy in the air whenever it is discussed in public, and they deserve a direct acknowledgement. For a long time, the claim that Jews had no ancestral connection to the land of Israel was used as a cudgel in political arguments about the legitimacy of Jewish national claims. The Khazar hypothesis was often deployed as a club, a way of saying, you are not from here, you have no historical tie, your presence is a colonial imposition rather than a return. The genetic data from the past 2 decades directly refutes the biological premise of that argument. Ashkenazi Jews share measurable ancestry with the Bronze Age inhabitants of the Levant, and that ancestry is not a trace amount. It is a major component of their genomes. The people who built the Canaanite city-states and later emerged as the Israelites are, in a literal biological sense, the ancestors of the modern Ashkenazi Jewish population. At the same time, the data also refuses to cooperate with any claim of racial purity or exclusive descent. The Ashkenazi genome is a testament to mixture. It proves that Jewish identity has never been a closed genetic system, that the boundaries of the community have been permeable across centuries, and that ancestry is never a simple binary. The science provides no ammunition for racism, for ethno-supremacy, or for simplistic origin stories of any stripe. It provides a nuanced, empirically grounded, and deeply human account of how a people emerged from a specific corner of the ancient world and carried that biological inheritance across 2 millennia of migration, mixture, and survival. And there is one more layer to unpeel, one more twist of the genetic kaleidoscope that reveals a pattern no one predicted. This is the moment, around minute 22, where we have to go back to the priestly Y chromosome. Because the Cohen modal haplotype does not just link Ashkenazi and Sephardic Kohanim. It links them both to an African clan with an extraordinary oral tradition. The Lemba people live primarily in South Africa and Zimbabwe. They practice customs that include male circumcision, dietary restrictions, and a reverence for a single deity. For generations, their elders have told a story of ancestral migration from a place called Senna, somewhere in the north, a journey that brought them across the African continent carrying traditions that echo Jewish practice. The Buba clan within the Lemba holds a special priestly status, and they claim descent from ancient Jewish ancestors. Western observers dismissed these claims for decades as fanciful, the product of colonial contact or missionary influence. Then the geneticists arrived. In the late 1990s and early 2000s, teams led by Mark Thomas and others tested the Y chromosomes of Lemba men. The results were astonishing. The Lemba as a whole carry a significant proportion of Y chromosome lineages that trace to the Middle East. But when the researchers focused specifically on the Buba clan, the priestly lineage, they found something even more specific. Roughly 50% of the Buba men carried the Cohen modal haplotype, the same Y chromosome signature found in Ashkenazi and Sephardic Kohanim, who trace their descent to the biblical Aaron. Read that number again. Across thousands of miles and thousands of years, the same rare Y chromosome combination connects Jewish priests in New York and Jerusalem to the priestly clan of an African people in the Limpopo region. The chances of this occurring by random coincidence are infinitesimally small. The genome is telling a unified story, a story of a specific paternal lineage that originated in the ancient Near East and then branched, carried by human migrations into Europe and across Africa, preserved in male lines that remembered their ancestry, and passed it down from father to son for perhaps more than 100 generations. Try to imagine the human scale of that journey. Picture a group of traders or refugees, perhaps several hundred strong, leaving the Levant more than 2,000 years ago. For reasons lost to history, one branch of this ancestral population turned west, following the Mediterranean coastline toward Rome and eventually the forests of the Rhineland. Another branch turned south, traveling perhaps down the Arabian Peninsula or across the narrow strait at the mouth of the Red Sea into East Africa, then pushing steadily inland over centuries, intermarrying with local populations, adapting to new ecosystems, new languages, new dangers. The cultural memory of the journey west became what we know as Ashkenazi Judaism. The cultural memory of the journey south became what we know as the Lemba oral tradition. The biological memory of both journeys is preserved in identical stretches of DNA on the Y chromosome of their male descendants. This is the kind of discovery that makes the hair on the back of your neck stand up. It connects the human family in a way that no ancient text, no legend, no act of faith could accomplish alone. The DNA does not care about the names we give ourselves or the borders we draw. It simply records descent, faithfully, and when we learn to read it, we discover that we are all carrying stories inside us that we have not yet fully understood. Now bring the earlier thread back, because it connects to everything you have just heard. The Ashkenazi bottleneck, that tiny founding population of roughly 350 individuals in medieval Central Europe, did not emerge from nowhere. Those 350 were the descendants of the mixed Levantine Italian population that had coalesced in Roman-era Southern Europe. They carried the Cohen modal haplotype in some of their male lines. They carried European mitochondrial DNA in the vast majority of their female lines. They carried autosomal ancestry that was already half Middle Eastern and half European before the bottleneck even began. When the population crashed down to 350 people, that specific mixture was what got through the bottleneck. The genetic diseases that later became hallmarks of Ashkenazi ancestry, the Tay-Sachs and the Gaucher and the BRCA mutations, were not ancient Israelite mutations. They were chance variants that happened to be present in the small bottleneck population, and they were amplified through generations because the effective breeding pool was so restricted. The bottleneck compressed the genetic variation of the entire diaspora into a tiny genetic snapshot, and then that snapshot was photocopied 10 million times. If you are Ashkenazi and you carry one of these mutations, you are carrying the physical evidence of a specific demographic catastrophe that befell your medieval ancestors. You are also, in a strange way, carrying the proof that your ancestors survived it. The genomic history of the Ashkenazi Jewish people is not, in the end, a comfortable story for anyone who wants a simple slogan. It defies the anti-Semitic fantasies of those who would sever the Jewish connection to the land of Israel by asserting Khazar descent. That fantasy is now scientifically dead. It also defies any narrative of pure racial descent unaltered by time and mixture. The Ashkenazi genome is a mosaic, and it is precisely that mosaic quality that makes it such a powerful testament to real human history. The history is not just in books. It is inside you. If you are an Ashkenazi Jew watching this, some version of this entire story is sitting in your genome right now. The Bronze Age Canaanite farmer in your DNA. The Roman-era Italian woman in your mitochondria. The medieval Rhineland founder who survived the bottleneck. The Polish-Lithuanian great-great-grandparent who passed down a BRCA mutation that a genetic counselor in the 21st century will screen for at a routine appointment. The Cohen Y chromosome that links a stockbroker in Manhattan to a priest standing before the menorah in the Second Temple. None of these things are metaphors. They are base pairs, A and T and C and G, strung together on chromosomes, faithfully copied through meiosis and fertilization, passed down in an unbroken chain from the deep past to this very moment. If you have made it this far, leave one word in the comments capturing how this makes you feel. Just one word. The textbooks have not fully caught up to this science yet. The history most people learned in school, or absorbed from the culture, still tends to treat ancient Israel as a vanished world, a civilization whose genetic legacy was somehow erased or irrecoverable. But the DNA tells a different story. The people of the Bronze Age Levant are not gone. Their descendants are alive today, walking the streets of Tel Aviv and Brooklyn and Buenos Aires and Johannesburg, carrying their genetic inheritance forward into a world those ancient farmers could never have imagined. The diaspora that scattered a people for 2,000 years across continents and oceans did not scatter them in the genome. The genome converges back to one place, to one time, to one small people who lived on a narrow strip of coastal Levantine land between the Mediterranean and the desert, and who wrote texts that would reshape the religious imagination of half of humanity. Their biological children are still here. 350 individuals in a medieval village 700 years ago, 10 to 11 million descendants today, and an ancient DNA revolution that has, molecule by molecule, brought the hidden history of a people into the light. Subscribe. We go this deep every time, past the comfortable version of the story and into what the DNA actually shows.